welcome all the attendees and our speakers for this rare disease genomics perspective talk. And this is like a series of events which we are conducting so that we can spread the education of genomics and how it is being used in clinical space as well. And rare disease is one of them. And rare disease, if I just gone through some of those statistics, is not so rare. That is a word that is now we can say 300 million people across the globe are suffering from this disease, and in India itself is close to 70 to 90 million people. And the real challenge is, we can say, diagnosis. And after diagnosis, the treatment is also sometimes only 5% people, or we have like only 5% diseases we are able to manage with medication. And rest of the stuff or the diseases, we are not finding any cure, but the research is going on. So I can have the next slide. So today's speaker, we have Dr. Ratna Puri. She is from Sir Ganga Ram Hospital and the chairperson of Institute of Medical Genetics and Genomics. And like the people who don't know, but in this, just I would like to introduce her in detail. But she's a very renowned person in the area of genomic genetics and genomics, and in terms of like she has awards, which is like IC Verma Outstanding Research Award in 19. She also received a Dharma Veer Award of Excellence in 2010 and a Young Investigator Award from Tokyo, Japan, 2006. She also member of many prestigious bodies in terms of rare diseases, I can say. So here we have listed few of them, which is Undiagnosed Disease Network, International, NIH, USA, and then we have another like International Rare Disease Consortium, the Diagnosis Scientific Committee member, and also she is the expert panel in the WHO Collective Global Network for the rare diseases and the Rare Disease International. And she is like ongoing projects and also completed, have many projects also. Well, few of them we have like Indian Undiagnosed Disease Program, the establishment registry also for the rare and the potential treatable genetic disorders, the molecular characterization of lethal skeletal dysplasties, and also in some muscular dystrophy diseases, which like a deep phenotyping and molecular studies is being conducted by her lab. And definitely the biggest tool that is now coming up in this rare disease diagnosis, the next generation sequencing. So they are utilizing this to get the answers for the intellectual disabilities. Altogether, more than 140 publication, I can say, and the things are growing up as the genomics field is maturing. We are also getting answers for all the unsolved questions. So I would request Dr. Ratna Puri to start her talk.
Thank you very much, Dr. Sharma, for the very kind introduction. And without wasting any more time, I'm going to share my screen. Is it seen and can you hear me clearly?
Yes, ma'am, we can see and hear you clearly.
Good evening, everyone, and thank you for joining in today on a working day early in the evening. And I'm going to talk today on something that's really close to my heart, and that's really addressing the whole scope of rare diseases and utilizing genomics in the clinic. So, you know, people know very little about genomics. We think disorders are rare, but actually they are not rare. They are just undiagnosed. Because all our lives, we think that genetic disorders diagnosis is unessential, it's all nebulous. But through this series of slides and case presentations, I'm going to show that how genomics is impacting the lives for patients in the clinic and how we are able to use it on a day-to-day to be able to assist these families in their quest, not only for a diagnosis, but also for management of their patients. So because I'm talking to a public at large, I think it's imperative to start with what the DNA is. It's all the genetic information that each living person, human or non-human, is made of. It is in the humans a 6-foot long strand that has 6 billion letter codes. And this double helical structure, as you see it on my screen, is the code of life. And the double helix is made of alternating ATGC, which are the 6 billion individual building blocks. And when we read them together, as we now have been after the Human Genome Project, we are able to understand what is the reference sequence of the genes. We are able to sequence this entire genome exome quickly. And over the last decade, we have improved computational abilities to be able to diagnose and identify disorders. So to put it very simply, genetics is this book of life. The individual pages are the chromosomes, and the words within this are the genes, and each alphabet is the alphabets of the DNA, which are the ATGC. And we can study this entire thing in a sample, which can either be a blood sample or a tissue sample. Either it's from a fetus or from an adult or a child. And from each of these samples, DNA can be extracted. Now, there is a host of genomic technologies which look at disorders of chromosomes as well as disorders of genes. And the tools have evolved very rapidly over the last 2 decades. So I'm just going to talk a little bit on this slide before I go ahead. Now, the first 2 bars are really looking at disorders of chromosomes. We are all familiar with the standard karyotype, which looks at the chromosomes at a level of about 5 to 10 MB. If we want to look at smaller specific points on the chromosome, we use fluorescent in situ hybridization, like we do for Williams syndrome or 22q microdeletion. When we are looking at, we want to look at the chromosomes in greater depth. You can see that the resolution is going on increasing. We use microarrays, which can either be array CGH or single nucleotide arrays. But to put it simply, we use the microarray to look at chromosomes at a very high resolution, as it's looking at something with a magnifying glass. Then we also know the new technology of non-invasive prenatal testing that is used to look for common chromosomal abnormalities of number involving chromosomes 13, 18, 21, and sex chromosomes. Beyond that, we come to disorders which are techniques that can also be used for chromosomes, like the MLPA, which is the multiplex ligation probe amplification. And this is but predominantly a technique that's used for single gene disorders. And we also have real-time PCR. Now here we go at exon level and single gene levels. Then we have Sanger sequencing, where each alphabet or each ATGC can be sequenced. We also have specific kinds of PCR for specific kinds of disorders, which I'll be talking about in my slide. And this is the repeat primed PCR or the TP-PCR. And then we have the new technology. And I think lots of us, because of the availability and the promises of this technology, feel that this is the answer to everything and this is all redundant. But at the beginning of my talk, I feel that this is as important as this, and this latter NGS is not the answer to everything. So next generation sequencing is looking at everything in a whole. And I think I'll leave it at that there, because I will address that in my subsequent slides. So when we talk about this technology, what is the application in the clinic? We can use it at various times in life. It can be used in the preconception period, either in the antenatal or in pre-implantation genetic testing. We can use it in the newborn. And before we come to the newborn, we can use it as NIPT or NIPS, which is a cell-free fetal DNA in the maternal blood. We can do RH testing on this, and we can do common aneuploidy testing of this. Then we come in the newborn period. either in a sick neonate who's not well or who's symptomatic, or in some countries in the world, they've also started using genomic technology as a part of newborn screening. However, we are not there as yet in India. Then we talk about predictive testing, carrier testing, post-symptomatic diagnostic testing. And this and tumor testing is the largest field in India where this technology of genomic is used. And what's the need to make of a diagnosis? I remember when we were studying in MBBS as well as during MD, we were told genetic disorders, no need to make a diagnosis. Why do we need a diagnosis? Because we know that most of them present with common phenotypes, which could be genetic or non-genetic. So a specific diagnosis is really important because for the family, it gives an answer. There's a relief from the uncertainty of knowing what is wrong with the child, an end to a very, very long diagnostic odyssey where they move from hospital to hospital and doctor to doctor to understand why their child is different and not developing as he or she should. We now know that there are specific therapeutic interventions for a large number of genetic disorders, and I will allude to this also in my subsequent slide. And finally, and actually very, very important, is genetic counseling for recurrence risk, future reproductive options, and the option of prenatal diagnosis to prevent recurrence of a disabling disorder. If we don't have a specific etiological definite genetic diagnosis, we are not able to provide any of these options. So through a series of slides, I'm going to show you the use of different kinds of technology, depending upon what the patient has and what is the appropriate test in that situation to do. So this is a child who presents with splenohepatomegaly and failure to thrive. The commonest disorder we look for in our country is beta thalassemia. So that is the first thing that's excluded. There were no infections and there is no malignancy. To rule out malignancy, a bone marrow biopsy was done and storage cells were identified that moved towards the diagnosis of a lysosomal storage disorder. An older child with a similar phenotype additionally can present with oculomotor apraxia. And in both these situations, we know that this is the predominant phenotype of a common lysosomal storage disorder, which is called Gaucher disease. So once you've made a clinical diagnosis that it could either be Gaucher or Niemann-Pick, you do certain screening tests, which are biomarkers, which alert you towards the diagnosis of those disorders. And in the blood, we do chitotriosidase, which is available in our country. Lyso-Gb1, which is very specific for Gaucher, is still not available. Once we find that the chitotriosidase is elevated, however, I must say here that in some situations, in about 10 to 20% of cases, the chito levels may not be elevated. We would do specific enzyme testing for Gaucher disease, which is done on a filter paper sample or in a heparin sample. And once we identify that the level of acid beta glucosidase is decreased, we go ahead for gene testing in a EDTA sample. So this is the chronology which is to be followed to be able to give a definitive diagnosis. And then to start the child on multidisciplinary management and definitive therapy with enzyme replacement of the deficient enzyme. Let's go to the second phenotype. In this case, this was a 2.5-year-old child, second born to non-consanguineous parents, and they had lost the first child with similar complaints, but without a diagnosis. Now, this child actually came to medical attention for increased stool frequency. So he was evaluated by a gastroenterologist. There was failure to thrive. But what the geneticist I picked up in this child was he looked a little different and the hair did not look okay. He did not have so much of protein energy malnutrition, which could explain this hair. So what we did was we plucked the hair, saw it under a microscope, and identified that this was trichohexis nodosa, which is a phenotype that we can see in some genetic disorders. And putting this entire phenotype of diarrhea, he had altered liver functions, failure to thrive, and trichohexis, we came to a clinical diagnosis of trichohepatoenteric syndrome. It's not a common syndrome. We had not seen a child with this syndrome before, seeing this one. But just by taking a good history and a very key examination, we were able to make a clinical diagnosis. Now, this was in the era pre-NGS. And when we went ahead to identify that is the genetic basis known and how do we do the gene mutation study, we did a literature review. A gene had recently been identified just a few months ago, but it was a very, very big gene. It had 43 parts, and it was very difficult to test this gene in those days. However, while we were reviewing this paper, in the paper, we identified 2 patients of Indian origin who both carried the same mutation. So we wondered, should we try this in our patient? So we just set up this one mutation in this child and confirmed that this child also had the same mutation. And subsequently, we identified another child with the same mutation. And most likely that this is a founder mutation in the Indian population for a not so common disorder, or at least a common Asian mutation for a not so common disorder that is called trichohepatoenteric syndrome. So as you can see within all this, that the phenotype of the patient and putting it in a logical and a systematic manner is really crucial to be able to give a diagnosis. Now, moving away, I come to the other technologies, which are the newer technologies. We have microarray for chromosomal disorders, and in a huge meta-analysis, the diagnostic yield of this beyond karyotype is 10%. And when we come to exome sequencing and genome sequencing, the yield increases to about 30 to 40%. Now, this was a child who was, again, second born with a cardiac malformation. And the first child of that family also had a conotruncal anomaly and had died without a diagnosis. This child also had a conotruncal anomaly, but that was different from the first child. But when you see 2 children with the same abnormality, you again have to think, is this a genetic disorder? The karyotype was done and it did not identify any abnormality. So we wanted to look into the chromosomes in greater depth and therefore did a chromosomal microarray. This identified a 22q microdeletion, which is something that cannot be picked up on a karyotype because it is small. It is too small to be picked up by the resolution of the karyotype. And we did this in the parents as well, but in the parents, we did not do a microarray. We did a cheaper test that is called fluorescent in situ hybridization. And we identified the mother to be a carrier of the 22q microdeletion. 10% of parents born to children with 22q microdeletion can be carriers. Although she was absolutely asymptomatic, she did not have any cardiac malformations, but this is well known for autosomal dominant disorders that they can be variable penetrance. So therefore now we had an answer for the family. We understood why their children had cardiac malformations and why it was recurrent, and how in the next pregnancy we can provide prenatal diagnosis at 11 weeks of gestation by doing a microarray and confirming the 22q, the presence or the absence of the 22q microdeletion. This is a common phenotype. It's a clinical diagnosis of Duchenne muscular dystrophy. And I think every clinician, pediatrician, neurologist, adult physician would in their career have seen a child with DMD or Becker muscular dystrophy, which is a milder spectrum. So what is the need of genetic testing for a child with a clinical diagnosis? One, there are many lookalikes like other limb girdle muscular dystrophies, and therefore you have to confirm the diagnosis. Because this is an X-linked disorder and you need to prevent the recurrence of this disease in other members of the family. The presence of a definitive diagnosis by the mutation testing can also inform the carrier status of other women in the family who could be at risk of having a baby with DMD. And today we have specific treatments, and I'll come to this here. So what is the test do we do? Do we do exome sequencing? No. We first do the MLPA that I've alluded to before. because 80% of the mutations in patients with DMD are because of deletions and duplications in the dystrophin gene. If you don't find a mutation by MLPA, then you go and sequence the gene. And here, because it's a very large gene and it's got 79 exons, or it's got 79 parts, you do a clinical exome or an exome sequencing, because that's a cost-effective technology. Why would I want to know what the specific mutation is? Because we have three kinds of treatment for patients with DMD. If there is a deletion that is amenable to exon 51 skipping, that means if I remove this exon 51, then supposing the patient has a deletion of exon 50, and I remove exon 51, 49 and 52 will again align well, resulting in a milder phenotype. So we do know that 13% of patients with DMD are amenable to exon 51 skipping. And the same is for exon 53, and now there is exon 45. And also we have some treatments for point mutations or termination mutations. That is the read-through. So therefore, until you have these mutations, you cannot even think about treating these patients. This is not an uncommon phenotype. The commonest inherited cause of X-linked muscular dystrophy, called Fragile X mental retardation, they present with a very common phenotype of developmental delay, speech and language delay, including some hyperactive behavior. It is a norm in the clinic to test every male with ID and a normal size or a large head. What test are we going to do? Are we going to do an MLPA? No. Are we going to do a karyotype? Not anymore in this day and era, in this day and age. Are we going to do a next generation sequencing? No. We need to do a specific test that is called TP-PCR, or the triple repeat primed PCR, because this is a triple repeat disorder, and it is beyond this talk to talk about what a triple repeat disorder is, because it is only this technology that will be able to differentiate a normal chromosome related to Fragile X compared to a child who is affected. So therefore, you do need a clinical diagnosis to be able to decide which technology to use. And finally, I come to the next generation sequencing, which has really taken the world for, it's really taken over all forms of genomic testing world over. Next generation sequencing really means being able to test multiple genes together within a platform. We do know that we have 20,000 genes. Now, if I test all 20,000 genes, it's called exome sequencing. It's ideal to do trio, that is parents and child, but it's too expensive. Predominantly, we do solo. When do we use exome sequencing? Where there is a likely genetic basis, but I'm not sure which is the gene I need to test. I can do a lower end test, that is a clinical exome, where I do have a specific phenotype, but that phenotype is associated with multiple genes, and I cannot test one gene one after the other because that's too expensive. So I use a platform where all the genes related to my phenotype are placed and I test them. And a clinical exome tests all known genes that are associated with disease. You know why the difference between these two is the rest of the genes, we don't know what role they have. So therefore, that is not covered in the clinical exome. And lastly, we have something that is called the whole genome. Now, if this apple is the whole genome, the exome is just about 2% of this. It's just a slice of the apple. We still don't understand what the meaning of the rest of the apple is. And that now we are moving towards whole genome sequencing in the project that I do in my department of the undiagnosed diseases, where we've used all these technologies. We've not found an etiology, but we are very sure that the child does have a genetic disorder. And that's where we apply whole genome sequencing to be able to try and ascertain a diagnosis for that patient and the family. Let me tell you at the outset, analyzing all this huge amount of data is not easy. It's trying to find a needle in a haystack, and therefore we really have to know which test we are doing and why we are doing it. Okay, so this is a family, this is a series of different children. The clinical diagnosis of a rasopathy, which is, something within the Noonan syndrome pathway. We know that there are 18 genes that can cause the phenotype. Of course, every child will have a mutation in a different gene. So here I can use a clinical exome or a whole exome to test the genes that can be associated with this phenotype and therefore be able to give a definite diagnosis to the patient. On the other hand, I have a phenotype where I'm really not sure of a clinical diagnosis, because here the child has seizures, there is regression, there is spasticity, so it's upper motor neuron. So many tests have been done. General tests, including specific genetic screening tests, karyotype, GCMS, a lot of enzyme analysis for metachromatic leukodystrophy, Krabbe disease, neuronal ceroid lipofuscinosis, Tay-Sachs, these are all normal. The NCV, VEP, BERA, fundus did not give any clue. This was the child who had absolute neuroregression by 8 years of age. He had lost all attained milestone. He could not feed, so he was on NG feed. And the MRI just showed some non-specific atrophy with periventricular white matter changes. In a situation like this, where the phenotype suggests a genetic disorder, but we are not sure what we are dealing with, we do an exome sequencing. And this is how, when we analyze the data, this is how we create an IGV. And this exome sequencing confirmed the diagnosis of a juvenile onset GM1 gangliosidosis in this child because there were biallelic mutations in the GLBL gene. Now, these mutations were pathogenic, they were reported and consistent with the phenotype. So this is how this technology really helps you. We also now identify various complex phenotype. For example, in this family, there were 3 people who were short and had skeletal changes. However, only this child had associated dysmorphism and looked different. When we did the exome sequencing, we identified first a gene that could be responsible for the skeletal abnormalities. However, this did not explain the phenotype of this child. And so further evaluation of the data of this child identified a second disorder because of a mutation in the DVL1 gene that causes Robinow syndrome. And the reason we looked into this data, because our first clinical diagnosis for this child was Robinow syndrome. And we thought because it's a dominant disorder, the father and the sibling had a milder version for the disorder. But on the first go, the report did not give us this Robinow syndrome. And it was only because we were so sure of a clinical diagnosis that the data was reanalyzed to be able to confirm our clinical suspicion. And as we see more and more patients, we realize that there is an entity now which is referred to as a dual diagnosis. And this is what this child had, a dual diagnosis, one gene causing the skeletal phenotype, which was inherited in an autosomal dominant fashion, and another gene that causes Robinow syndrome, which was de novo in this child. And I think this is my final case where we now are moving and doing preconception carrier screening. However, this is a very gray area and needs to be dealt with a lot of caution and analyzed with a great deal of care. The case that I'm showing you is something that is very specific because I just go to show you how we can use this in the clinic to help the families. But it's not so easy for all patients. The reason we did carrier screening for this family were they were consanguineously married and their first child had galactosemia. So they wanted to plan another pregnancy, and because they were consanguineous, they felt that they could be carriers for some other genes, and they wanted to ascertain the risk before they planned another pregnancy. And when we did the carrier screening, we got a host of genes for which both partners were carrier. You can see the one with the star is the only one amongst this that was reported in literature previously. But when we saw the other ones, the nature of the variations was such that they were significant and pathogenic. And the reason for that was, now the GALT gene, which was also identified in the variation was also identified in the child, was the same. So, and because we had phenotyped the child and done the enzyme, we could convert this into a likely pathogenic. And this gene and the primary hyperoxaluria gene are linked. They are on the same chromosome. And therefore, this was inherited and linked as a whole. And the child also had phenotype of renal disease consistent with hyperoxaluria. And additionally, there were also carriers of two other unrelated disorders, which was Ehlers-Danlos and xeroderma pigmentosa, both of which were significant. The Vicky syndrome was uncertain significance, and we did not take that into consideration for counseling this family. So when we see this family, the challenge for them is that you have to test for actually 4 disorders. If there is galactosemia, they would have inherited, the child would inherit primary hyperoxaluria gene also. So there were really 3 groups of disorders for which the fetus was at risk. So in view of this, they planned an IVF with an egg or a sperm donor. They offered for an egg donor in view of the high genetic risk. And here we know that because there are so many disorders for which they are carrier, the option of being able to test the donor for the carrier status of these specific disorders could be offered to the family. So to conclude, when you have patients with a suspected genetic disorder and you confirm the disorder by any of the technologies that I have discussed, you either can give them definitive management, and here we have a large number of inborn errors of metabolism, the channelopathy seizures where we can alter the drugs that we can give to these patients, spinal muscular atrophy, DMD, Prader-Willi syndrome, Fragile X syndrome. And I've left this because there's a host of other disorders, all of which I cannot enter here, that are now amenable to definitive management. If not, we also have a whole lot of supportive therapy and other things that can influence management. And on the other side, we would know that, well, for this disorder, there is no effective therapeutic intervention, and we could counsel the family about the prognosis. And of course, as I've discussed with you, we are coming into an era of preconception carrier screening, but here I would like to address this has to be used with a lot of caution. And possibly in October, the Indian Academy of Medical Genetics is going to do a small meeting that is related to preconception carrier screening. And I would request you all to be alerted to the emails that you receive for this session. So to conclude, we have a lot of access to technology that is not so expensive as it used to be in the years gone by. But we also understand that one jacket does not fit all. And therefore, it is important to understand what each genomic test can offer, because we know that a definite diagnosis can influence patient management. It is very important to appreciate that to be able to identify a causal gene variant, you need precision of phenotype. And if you don't give good phenotype, you may not identify the variant that you're looking for. And because the bioinformatic analysis has to correlate with the phenotype. So it starts with how we started medical school, with a good history, a family history, a head-to-toe examination, putting everything together to create a thought process, creating a differential diagnosis. And if you do it in the systematic manner, you can optimize the great utility of this new technology. And therefore, I feel that we are now in an era where teamwork and multidisciplinary care is really important, because one cannot do it all. So let's not be taken in by just this whole testing that is all over the place. I think it's important to do it in a very, very systematic manner so that we are not overwhelmed by the data. Thank you very much.
Thank you, Dr. Puri, for such a wonderful talk. I can say you have covered most of the aspects that are associated with rare diseases, their diagnosis, their prognosis, their treatment options, what are the challenges right now people are facing. And yes, definitely economics is one of those stuff that is the burden on the society is also challenging when we are not able to manage the rare diseases. And with these out of like some diseases we have answers, we have a proper diagnosis, but sometimes we don't get. And we got some examples from a genomics also where there was no answer from their medical field, but when they did the genomic testing they got a new answer, and they were treatable also. So finding a treatment is something that is luck still, I will say, whether that mutation or whatever inference we are getting from a patient, whether a treatment is available or not. And definitely disease management is one thing that people are focusing on, how to develop drugs for that, how to formulate drugs, how to give nutrition to the people who are suffering from their diseases, because their nutrition sometimes is very, very different from the routine people. So it's like a very enlightening talk, I can say. I think we will be having a question and answer session, and in which we will be taking up some of those questions, and also some panel discussion will be going on after the talk of Dr. Harsh Sheth. So now I would like to introduce Dr. Harsh. Uh, he is assistant professor and head of Advanced Genomics Technology Division at FRIGE Institute of Human Genetics in Ahmedabad. I know him personally. I interacted with him, and definitely I can say he's one of the person who really working on these new technologies so that it can be made cost effective, and definitely in which he had got success also, especially microsatellite instability studies and small molecular probes based SNVs and the inversion probe CNVs detection. The area of expertise from we can say completely, I can say NGS plus computational biology, and mostly working in the area of cancer prevention, male infertility, and rare diseases, and definitely molecular and epidemiological and the personalized medicine. And membership, I can say our list is huge, but we have figured out, but mostly he's associated with all those all those body of human genomics, I can say he's associated with. Apart from this, he got received professional honors from South African Open Genome Project, and he's a member of the ICMR subcommittee on precision oncology. And publication are, we can say, started off like 30 peer publications are there, book chapters his group has written on the cancer prevention and lysosomal storage disorders. And research grants, definitely there are a number of grants that he has received or his lab has received. People have like multiple PhDs also. He has like from his lab has been completing his PhD. Apart from this, the main is like focus of Dr. Harsh will be like how he has used his technologies to make a cost-effective way of putting the diagnosis for the we can say affordable at the same time. And also he is an adjunct lecturer at Gujarat Technological University, Ahmedabad. So with these words, I would like to welcome Dr. Harsh for his talk.
Thank you, Dr. Sharma, for that wonderful and generous introduction. Of course, Dr. Ratna Puri, she gave a wonderful blizzard of information, all the way from how to diagnose and what is critically very important to recognize the phenotypes and then use the right set of technologies, because sometimes some techniques don't work in a given set of diseases. But in fact, I like to take your attention to her very last slide, whereby she says that data can break your computer, whereas in my case, Zoom has managed to break my computer. That is why, annoyingly, I have to give it from my iPad. So please bear with me whilst I find a way to
share the screen.
Yeah. I don't know if you're able to see my screen. Is it visible?
Yeah, it is visible on the full screen. Yeah, we can see the presentation also in a normal mode. If you can do that.
All right.
It's coming up. Yeah.
Yep. All right. Hallelujah. So let's see if we can play around with this technology. So pardon me for this. Essentially, what I want to talk about today is, whilst that all of these new technologies have come up, chiefly your whole exome or even whole genome, they're still very, very expensive. I mean, you're talking in the ballpark of anywhere from 20,000 going all the way up to 80,000. Now, an average Joe in India won't be able to afford a 20,000 rupee diagnosis, especially in rare diseases scenario. So how can we start working towards developing an indigenous genomic technology that can help not only in diagnosing these patients, but later on it can help in preventing these diseases in the first place? So I'm going to talk about two very simple diseases. One is lysosomal storage disorder, the other one is Lynch syndrome, and both of them are very close to my heart. And both of these diseases came to my attention. Well, Lynch syndrome came when I was studying in the UK, and lysosomal storage disorder when I came to my attention when I moved back to India way back in late 2018. And throughout my entire work span, so as Dr. Sharma very nicely informed you all, I worked all the way from pharmacogenetics of warfarin to developing rapid molecular diagnostics. Let's see. Why is it misbehaving? I'm so sorry. It somehow doesn't seem to like it. There you go.
Yeah.
I'm currently working on the role of de novo mutation and identifying new genes associated with male infertility. I've even worked on cancer prevention, especially colorectal cancer prevention, about which I'm going to talk to you today, and a whole host of other stuff that my lab is currently working on, anywhere from autism spectrum disorder to National LSD Biobank, and so on and so forth. So the whole crux of my entire talk is actually summarized in this one line, which was written by Thomas Adams in 1618, which states that he is a better physician that keeps diseases off us than he that cures them being on us. Prevention is so much better than healing because it saves the labor of being sick. And this one line, this one sentence is extremely pronounced when it comes to the world of genetics, because it's far more better to prevent the disease from coming in the first place, rather than worrying about diagnosis or treating it. So let's start with lysosomal storage disorder. So many of you may know that these are a group of 70 disorders where the lysosomes, which are small bodies inside our cell, they're essentially kachala. Essentially, they collect all the stuff that all the garbage that is produced by our cell, and they help in processing that garbage. But that lysosomal function is disrupted. And these diseases follow two modes of inheritance. It's either autosomal recessive, that means both copies of your DNA is defective, or it's an X-linked model. That means only one copy of X, especially in male. If it's defective, it can lead to the disease. The problem, however, is that you get a systemic collection of phenotypes. So that is to say that multiple diseases can have overlapping phenotype, thereby making clinical judgment rather difficult. Now, whilst these are rare diseases individually, but when you bring them together, the combined prevalence is 1 in 5,000 worldwide. So it's not so rare as one might think. Now, out of these 70 diseases, there are 20 which are about 23, which are very, very commonly observed in India, namely Gaucher's, Tay-Sachs, and Sandhoff. So the way you generally go around identifying these patients is, first of all, they would come to your clinic. You would look at, you would perform a differential diagnosis based on their clinical symptoms. You then do a biochemical screening so as to narrow down which set of LSDs you're working on. You would then want to carry out enzymatic confirmation, so you hit on the right LSD. And once you have that, you would want to carry out, oh, not this again. You would want to carry out sequencing to identify the causative variant, right? But it's not as easy as it sounds, because like I said, you can have overlapping phenotype. And because of that, sometimes you go into an iterative cycle whereby you would want to analyze the first disease. If that's not the case, then let's do the second one, and so on and so forth. And because of that, it is reported that the diagnostic odyssey in some patients can go all the way up to even 5 years since initial presentation. I'm sorry, in this day and age, that is just not good enough. Not only that, because of this iterative nature, the cost for the patient also increases substantially with each round of iteration. Furthermore, because it's a biochemical test, they're very, very hard to establish, and therefore it requires skilled manpower and a complex lab setup. So it's not amenable to high throughput. From one study from Belgium, they found that when they were doing biochemical testing, their yield was just 5%. And I'm sorry, but a 5% diagnostic yield is very, very low. Now, if you were to take that number and put it in an Indian context, it would mean that right now there would be 240,000 children with LSD, but only 12,000 of them would be known to the medical services. So that's an absolutely horrendous number. And what we require, therefore, is a high throughput, low cost, and easy to use diagnostic tool. Now, one might think, okay, what about sequencing? Let's just forget the whole thing. Let's just do sequencing. But it's not as easy as it sounds. So here is a typical example. So these are 3 different studies who use different types of panels of different sizes. So one was looking at 800 genes, the other one was looking at 60 or 80 odd genes. But look at their detection rate. It's about 50 to 60%. So it's not as good as it sounds on the tin. Not only that, we have a bit of a problem with NGS, because our own tendency is that the larger the NGS panel is, our anticipation is it would lead to an increase in diagnostic yield. But we just saw that with 70 gene panel, you got a 60% diagnostic yield. But when you scaled it up to 900 gene panel, you got a 7%, 60, 70% yield. So that's not a substantial increase in diagnostic yield. Plus, when you increase the size of the panel, you're also increasing the cost per sample, because, well, the instrument is, you're going to take so much space on the instrument to analyze that one sample. And because of increase in size of the panel, you would require a larger instrument, and therefore a larger infrastructure, and therefore more labor costs, and therefore the whole starts to snowball, and it becomes very, very expensive. Now, I think Premas would probably agree with me that Illumina MySec is the most common and most commonly found platform in India right now. Is that right, Dr. Sharma? Right?
Yes.
So clearly, our goal was to try and develop a panel that would fit on that platform, because then majority of the labs across India could use their instrument to diagnose these diseases. And not only that, rather than looking at all 70 LSDs, and of which only 23 are the most common and the rest are quite rare. So we focused on those 23 LSDs. To do that, we had a team, which Dr. Harsh Sheth and myself, we led. My PhD student, Adina Nair, she was involved in development of this panel, and majority of the sequencing work was actually carried out at Gujarat Biotech Research Center, which is led by Professor C.G. Joshi and Dr. Madhuri Joshi. And the idea was that you would cover 20 genes that are associated with these 23 common LSDs. And it would be a targeted panel. And this panel would be developed according to the ACMG guidelines. Now, ACMG has prescribed a wonderful set of guidelines, whereby if you were to design this panel and you were to hit all the marks, then it's quite likely that it has a good impact. So that's exactly what we did. We also made sure that it fits well within the limits of Illumina MySec flow cells, so majority of the people could use. And the most important part, not only should it be able to detect point mutations or single nucleotide variants, it should also be able to detect deletions or duplications accurately, because in these diseases, you do find them quite often. And because of all of this, it has to be low cost and amenable to high throughput. To do this, we went into the literature, and we found this wonderful paper, which describes a technique known as single molecule molecular immersion probe. Essentially, you just have a really long piece of DNA that sits on either side of the region of your interest that you want to sequence. It then makes a copy of that region, and then you can start sequencing it. Now, for our 20 genes, we developed 903 MIPS. Now, the thing about these probes is that the upfront cost of developing them is very, very, for 100 MIPS cost 12 lakh rupees, which at that time was probably the biggest order ever to come out from Gujarat for oligo manufacture. But each of these probes can be used for more than 1 million reactions. So now the cost per sample goes way below 1 rupee. So you can clearly imagine that it starts to become really, really cheap. And the pathway is also very, very simple. You carry out library prep that takes about 1 day. You do library pooling. That means you pool all the samples together. That takes about a couple of hours. You do the sequencing on the platform, and then you do computational analysis, whereby you call both point mutations as well as copy number variants, and then you go on to reporting. The whole thing takes a maximum of a week, end to end. Now, before we could really test this kit out, what we wanted to see was whether it was able to diagnose variants, which we had already identified. So we took 49 samples, and lo and behold, we got 98% concordance rate, which was pretty reasonable. Now, in one sample where we didn't find concordance was because that region belonged to a low complex, had a low complexity structure. That is to say, a single nucleotide may repeat itself multiple times, like AAAA. So MIP is not able to target such a region properly. So therefore, it was not covered, and that is why we missed that one variant. But this was something rather critical. Our assay was able to detect copy number variants. Now, here is an example of a multi-exon deletion that has occurred, and it's a heterozygous deletion. It means deletion is there only on one copy of the gene. But we also had the power to do a single exon deletion testing as well. So on the left-hand side, you can see that it's a homozygous deletion of 2 exons of the hexa gene. And on the right-hand side of the panel, you can see that it's a single exon heterozygous deletion of the hexa gene. So we knew that we were hitting all the right spots. But we wanted to be absolutely sure that when we put it out in clinical practice, how well the whole thing would work. So we took 175 patient samples, which were brought in clinically, which were sent by various doctors. And they were either clinically suspected to have an LSD, or they were previously diagnosed to have LSD via biochemical assay, but no genetic testing was carried out on them. So in these patients, we found that our diagnostic yield was 80%. That is to say, out of 175, we got diagnosis in 139 cases. And all of these variants were validated by Sanger sequencing in qPCR, they all turned out to be right, and we're very happy for that. However, there were a couple of diseases where our diagnostic yield was low. One was Gaucher's, and the other was Hunter. And the reason for that is that, again, you have the problem of either presence of a pseudogene or a low complexity sequence structure, because of which either your alignment fails or the probe doesn't cover that region at all. And it's not something that we have come across as well. It's something you would find in whole exome or even other targeted panels where these regions are missed out. So we were again hitting the right part. But remember, it's an 80% diagnostic yield, not 50%, not 60%, but 80%. Now, one might worry about the cost. Well, the cost of doing DNA extraction, library prep, sequencing, and even data analysis all pulled together is 10,000 rupees to the patient. If somebody comes and tells me, well, that's fine, but what if you come along with a VUS? Well, then we can do enzymatic confirmation as well on top, and the whole thing would then cost 12,000 rupees to the patient. And because we are using MySeq sequencing platform, We have flexibility of scaling. If we just have 4 samples, we can still run on MySeq. If you have 50 samples, we can still run it on that platform because you have multiple flow sets which you can use. And in terms of data analysis time, it takes 30 to 45 minutes per sample to go from FastQ to you having a list of variants for analysis. And this is not on some fancy server in the cloud. This is using a simple desktop, right, with a Core i5 processor, and it was able to do that. We wanted to keep it as simple as possible. And the whole thing, the whole turnaround time could be as little as a few days. Now, one might get slightly tempted to say, let's just do this assay across our entire patient cohort. But here is where the pinch point is. If there is a high suspicion or likelihood of a particular LSD and you feel that there is low risk of iterative testing, then the traditional approach whereby you do biochemical screening and enzymatic confirmation first might turn out to be a cheaper option. But in cases whereby you're not very sure which particular class of LSD this patient is sitting in, then you can do this particular assay first, do variant validation, enzymatic confirmation, and voila, you're good to go. Because there, at least you would have 80% chance of getting the right answer. In terms of public engagement, well, with this assay, we do a lot of regular online blogs or in-person discussion, just like this not only with clinicians, but patients and the general public. We also prepare a lot of brochures and pamphlets in English and regional languages, so that the patients can see whether my child has this set of phenotypes, then I want this tested as possible. In terms of data sharing, all the data that we have generated to date and more is now on European Nucleotide Archive, and all the variants are on ClinVar. So anybody who wants to play with the data, please feel free to go ahead, have a crack. And not only that, right now I'm in Freiburg in Germany, and my PhD student Adira, this was yesterday, she was presenting her poster. She won a travel award for that. So good on her. This was just a photograph from yesterday. So we go to scientific meetings as well to present. As a follow-up of this, so now that we have started diagnosing a lot of these patients, we want to understand the natural history of these diseases. So for that, Dr. Sheth just this year itself got a major funding from the Department of Biotechnology to create India's first biobank for these patients. And the idea is that you would hold DNA, plasma or serum, urine, and even biochemical and longitudinal clinical and anthropometric data for these patients, and you would on regular intervals follow these patients to see how they are progressing. As part of this biobank, we are doing this genetic test free of cost. So if you have any patients and you have very high clinical suspicion of LSD, you feel free to contact us, and we'll be very happy to do genetic testing for them. All we require is patients' clinical details and consent. And once the biobank is ready by 2024, all the data will be made to all investigators. And access to the samples for research will also be made available in time. So to conclude the first part of the story, the current gold standard technique may be iterative and is not amenable to high throughput, but our assay is able to look at 23 most common LSDs in India, and it's able to detect not only SNV, but CNV with a whopping 80% diagnostic yield. And because it's low cost and high throughput, it can lead to improvement in diagnostic yield. Therefore, you would have a higher chance of receiving treatment, genetic counseling, prenatal diagnosis, and therefore, most importantly, reduction in disease burden when you are planning for subsequent pregnancies. And again, you can think about flexible deployment approach. You don't have to go pure genetics, pure biochemical way. There's always the middle part which you can take. And as I said, the assay is now available for clinical use throughout India. So feel free to contact us, and we'll be very happy to help you out in providing diagnosis. Now, the second part of my story, I hope I'm pretty much on time. It will not take too long. Starts with Angelina Jolie when she created a massive wave in 2013 by saying that she's a BRCA1 mutation carrier, and therefore she's going for double mastectomy and oophorectomy. And after this one particular incident happened, there was a massive rush for for patients going to breast cancer clinics. It nearly doubled in the UK in 2014. Essentially, what she was trying to say was that if you are at risk of getting a disease, you can work towards reducing that risk via several interventions. Now, as we know, breast cancer, many other hereditary cancer syndromes follow what we remember from our school days as Knudsen-2 hypothesis. That is to say that when you are born, at least one copy of your DNA has a mutation in it. And therefore, you have a higher lifetime risk of getting a cancer, and that also at a younger age compared to people whose both copy of the DNA are absolutely intact. And therefore, they may get cancer during their lifetime, but they would most likely be older because both copies would have to be knocked down. So I want to take example of Lynch syndrome and most specifically colorectal cancer. And as clinicians in the audience may know, on the left-hand side, you can see this beautiful, smooth colon. And as you go along the neoplastic pathway, you end up with a small polyp, which then turns into adenocarcinoma, and then it goes towards metastasis. Now, when you look from the genetics perspective, there are two broad pathways to go into it. The one on the top is known as familial adenomatous polyposis pathway. Essentially, your DNA gets scrambled all around and you end up with cancer. The Lynch syndrome pathway, the one at the bottom, is whereby your spell checker genes are knocked out, and therefore you get a lot of point mutations occurring all over the place. Your DNA looks fine, but you get a lot of point mutations. And this is the one that I'm really interested in because it happens in every sick patient walking through the clinic. Now, this particular disease was named after this guy, Henry Lynch, who was in fact a boxer, then turned into a medical doctor. And he was the one who identified this disease. Previously, it was known as hereditary non-polyposis colorectal cancer. But later on, he showed that, no, it's not just colon, but you get a whole host of other types of cancers as well. You get endometrial cancer and upper GI and genitourinary cancers in these patients. And WHO estimates that the worldwide prevalence in the general population for this disease in 1 in 125. Now, this number is whopping because it makes it more common than even Down's syndrome. Now, if you find a carrier for this particular syndrome, then they have up to 70% lifetime risk of getting cancer, especially if MLH1 gene is knocked out. Now, this disease occurs because one of the 5 mismatch repair genes are knocked out, MLH1 being the most prominent one. And the way generally people would identify them is once somebody has gotten cancer, especially colorectal cancer, you would do MSI testing or immunohistochemistry testing. If that's normal, that's fine, most likely going to be sporadic. But if it's unstable, then you want to do BRAF testing to see whether they are likely to require germline testing. And if the germline testing comes out positive, then you're sure it's Lynch syndrome. But MSI testing is really not as simple as it looks. Because, and to give you some idea, when you're typing a text on your phone, right, sometimes you would make an error. Your phone would tell you that, hey, you've made an error, let me go and correct it, right? In the same way, when your DNA is making a copy of itself, there are parts of the DNA just like this, where the same letter repeats itself multiple times. And it can, and sometimes your polymerase can either add or remove one letter just by chance. So these mismatch repair proteins would come along, they would correct this error, and you would go home scot-free. But if this system breaks down, then you start accumulating errors in these regions. And this is what is known as microsatellite instability. Now, there are, like I said, there are only 2 techniques right now to detect it. One is immunohistochemistry, whereby you look at presence or absence of the 4 mismatch repair proteins. And the other one is fragment length analysis, which essentially looks at the length of these microsatellite regions to see if these regions in the DNA are different compared to what you would find DNA in your blood or other matched normal sample. The problem with techniques is that they require manual interpretation, which can lead to interoperator discordance, and therefore it's not amenable to high throughput. And because it requires so much manual intervention, the cost per sample is not affordable, especially in low income and low resource settings like India. They cost about 8,000 to 10,000 rupees in India right now. And because of that, it is estimated that fewer than 10% of Lynch syndrome patients are known to medical services worldwide. This is not just a problem of India, it's a worldwide problem. And again, just like LSD, there is a need for a high throughput, automatable, and cost-effective MSI assay. So we go back to our good old friend, molecular inversion probes, just like I showed in LSD. And there are two things that we did. We first identified a set of very short mononucleotide repeats. That means the same letter repeats itself multiple times. And what we wanted to see was whether we are able to find this deletion or insertion of one nucleotide or not. And lo and behold, we did find a set of 24 such markers that could do that. But that wasn't enough, because the polymerase can also make an error during PCR. So all of these microsatellites were right next to an informative SNP. So now if these insertions or deletion event only happened on one allele of your DNA, then you know for sure that it's a genuine a microsatellite instability. If it occurred on both copies of your DNA, you know that it's a PCR error, right? So based on that, based on that, we developed an assay that not only screens for these 24 markers, but we also added BRAF and KRAS on top as well. Because as you remember from my pathway diagram, you require BRAF testing, and KRAS is probably one of the most common gene mutations you want to screen for, because accordingly, certain therapies are given to these patients. So again, you would have the same workflow. You capture, you'd amplify, you'd prep, and you would sequence. But here's the trick. When it comes to data analysis, again, put data on your desktop PC, require no more than Core i3 or Core i5 processor, and you let it run overnight. And by the time you come in the morning, all the samples have been analyzed and it gives you a score. And if the score is above 0, it's microsatellite instability. If it's less than 0, it's microsatellite stable tumor. And we got 100% concordance with currently available clinical assays. So we're very, very happy. We got 2 worldwide patents on it. And right now we are working towards putting it in clinical tests. Now, the applicability, as I said, of an assay like this is multifold. It can help you to identify patients who are likely to have Lynch syndrome, what sort of treatment they should give, and whether they should be given immunotherapy, which is a very hot topic these days in oncology. But once you've identified the Lynch syndrome patients, that's not enough. We need to work towards preventing these cancers occurring in these patients in the first place. So here I go back to early 1990s, whereby at that time my supervisor, Sir John Byrne, he was working on the CAP2 trial. Essentially, he wanted to see whether aspirin or resistant starch is able to reduce risk of cancer in these patients. So he came up with a trial called CAP2, whereby he took 1,000 Lynch syndrome patients and divided half of them and gave half of them aspirin, half of them placebo, other half resistant starch and other half placebo. So essentially, this was a 2x2 factorial design, or 2 trials for the cost of 1. And in 2020, we published the longest follow-up on these patients, whereby these patients are given these aspirin intervention for 2 years, and we followed them up all the way up to 20 years. And at 20 years, we showed that the risk of having colorectal cancer was reduced by half in people who were given aspirin. Or to put it in simple numbers, the number needed to treat was 24, which means that if you have 24 patients to whom you give aspirin, you will save cancer at least in one of them, which is rather substantial. And here are the graphs of what I'm talking about. When you see this separation of the lines, what you're seeing is the one at the bottom is the aspirin, the one in the blue is placebo. And as you can see, the lines started to separate at 5 years. And when it came to 20 years, those lines were still far apart. That means that you have a sustained effect of aspirin all the way up to 20 years, which was excellent. And based on that, UK's National Institute of Health and Care Excellence put out a guideline recommendation saying that if you have a Lynch syndrome patient, you should prescribe low dose aspirin, that is 100 to 150 milligram of aspirin daily to reduce risk of colorectal cancer. But what about resistant starch? Well, just a month ago, that particular paper got published, and there we showed that patients who were receiving resistant starch, or who were randomized to resistant starch, had up to 50% reduction in risk of upper GI cancers. That is your stomach, your duodenum, bile duct, and pancreas. Now remember, these cancers are extremely hard to diagnose and extremely hard to treat. So if you're able to prevent them, that's a win-win. And the amount of resistant starch we gave them is equivalent to you eating 2 slightly raw bananas a day. So, if you have some free time and you're peckish, go for a banana. It will keep you healthy. And just like aspirin, these are the graphs whereby we are able to show that patients who were given resistant starch, from 5 years onward, these lines started to converge, and they remain separate all the way up to 20 years. Now, based on all of this data, I was then invited as a panel member for ICMR subcommittee on precision oncology. So all of this is now going into the latest guidance statement, which will be published hopefully either this year or early next year. Not only that, because we identified so many Lynch syndrome patients in India, we are now the largest submitter of prospective data on these patients on the PLSD database. So that's the worldwide database that helps you to formulate guidelines for these patients. So to summarize, MSI assay together with BRAF testing in colorectal cancer biopsy can help detect potential Lynch syndrome patients. And if you're thinking about preventing these cancers in this patient, you can either give them aspirin or resistant starch to reduce risk of either colorectal or upper GI cancers. Now, there are guidance available that says that you can give low dose aspirin if you do identify Lynch syndrome patients. And in terms of optimal dose, as to which dose of aspirin you should give, well, there is another trial ongoing right now, CAP3, and the data for that will come all the way in 2025. So stay tuned till that point. The minimum duration in terms of aspirin intake is recommended at about 2 years. Of course, there are some anecdotes that suggest that if you give them for longer duration, there is improved chemo prevention, but more data is required in that perspective. So lastly, I would like to thank you all for listening, and all the wonderful people that I've had the opportunity to work with, including our funders, GSBTN, DBT, Cancer Research UK, and so on and so forth. Thank you all. Thank you all for listening. And I'm sorry if I had any glitches in the middle.
Thank you, Dr. Harsh, for this wonderful technological talk, I can say, because what is in the background, how exactly the test is being developed, and what are the need of the assay, and how to design a very specific assay for a specific syndrome, and what things to be considered. So we covered the clinical aspects by Dr. Ratna, and the technological aspects from your end. And now we can start our panel discussion, and we can take up some questions. People can raise their hand, we can unmute them so that they can ask questions directly. So first of all, my first thing that I would like to open this panel discussion is like, we know that most of those rare diseases, there are 3 pillars for in which the things are clinical awareness is one stop, clinician awareness, I can say, and the skill set of the lab person, that is that, and the counselors. These 3 people are the important aspect where, because rare disease people go to the hospitals for their treatment per se. And we know every hospital is not being equipped by all these 3 solution at the same time, I can say, the people at the same time. So how do we expect them to come together in terms of networking, or in terms of, because skill set is sitting somewhere, the counselors are sitting somewhere, and the clinician is sitting somewhere. So, and rare diseases is everywhere. So how to bridge this gap all together, or what thoughts are there for both of you, I can say. I can take Dr. Ratna to speak first, and then Dr. Harsh can.
It's with programs like yours, trying to reach out to people to let them understand where the skill sets are and how to take forward a diagnosis. I think that's essential.
Yeah.
There are so many, now we have many centers all over the country. It's not as if they're just 1 or 2 centers. There are many centers, and so people don't have to travel the length and breadth of the country.
Yeah, true. The rare disease program has been established now by Government of India, and also strengthening the existing labs. That is also good initiative, I can say. And apart from the financial support, or the kind of support that is placed by Government of India, is good enough right now. Dr. Sheth, would you like to
So towards this, DBT actually took a wonderful initiative. It's called, they started with Nidan Kendra. So essentially,
Yeah.
they would pair a lab with a hospital, and so that they can start sharing each other's expertise. And such programs by the government are really coming to force right now, because they recognize that rare diseases, as we have demonstrated, they are not so rare, and it's far more economical to prevent such diseases from occurring in the first place than to worry about treatment. And I know SMA right now, people talk a lot about it and how expensive it is. If we worry about treating every single patient with SMA, it will probably take up our entire budget. So such programs by the government are really taking force now. Even programs led by Premas and many other organizations to not only connect doctors with within tier 1 cities, but also tier 2 and tier 3 villages. And the final layer, which I think, at least I know for sure, DD Gujarat or DD Girnar is doing, is they have Hello Doctor programs. So they do a lot of regional interactions on a whole variety of topics every week, which is excellent, because we can't be everywhere, but they help us to reach everywhere.
Yeah, thanks, Dr. Sheth. The other part is definitely the IT part, or the informatics part, I can say other. And I think Dr. Harsh has included this in his presentation, how like a desktop has been converted into a reporting machine where they can feed the data and the report can be generated. So that is one of those aspects. But yes, Dr. Ratna Puri also pointed out, when there is a huge data of genome and exome, then that becomes a very challenging to get the answer from that data set, and it takes a lot of effort. So I would like to comment on that aspect as such, like in as we represent Illumina in India, and Illumina globally also, solving this IT part, we can say the high throughput analysis per se for variant calling. We call it Dragon Server. So Dragon Server is able to deliver the variants in 20 minutes for genome and less than 5 minutes for exome. So that is a very super fast, and Guinness record has been established using this particular stuff for NICU patients, where the diagnosis was very, very important, so that a treatment can be initiated if there is a possible treatment. So this is part being taken up, but yes, that has to be again, we can say a setup has to be in-house, or if people want to use it on a cloud, that is also available. So things are coming up, and AI is one thing that I think I can say Dr. Harsh will be able to address that, how AI is going to change this whole scenario.
So, Dr. Sharma, what you say is absolutely right. There are so many companies coming out with a whole host of solutions. You have Google coming out, you have Amazon Web Service, and now Lenovo is coming into fray as well with their Ghost architecture. And even NVIDIA is coming into play. In fact, when you talked about artificial intelligence, it immediately hit me about NVIDIA, where it's not a CPU, but a collection of GPUs that all work together to analyze your data very, very quickly. The problem, however, is it's not data analysis by the computer where you'd find stagnation. At the end of the day, when you have those list of variants, somebody would have to sit down and manually curate them. Out there, to some extent, you can use artificial intelligence, that is to say, based on a certain set of phenotypes that you would find humans and zebrafish and mouse. Can we start prioritizing a list of variants? It's something that we at our institute use. But still, it's that manual intervention at the end, which is absolutely critical, and we cannot remove it in any way, shape, or form. And that is where you would find the real bottleneck is. So right now, if there are any biotechnologists sitting in the audience, budding biotechnologists, and are looking to find a field to work into, well, bioinformatics is really gaining attention, guys. And that is the field you might want to go into.
I'd like to chip in there. I'd like to chip in there. So we do have in my center the Ghost software, because I run a program which is the Undiagnosed Diseases Program. So we are doing our exomes and genomes. So in that, we are predominantly doing genomes for a cohort of patients where exomes hasn't given an answer. And very, very much like what Dr. Harsh said, that the computer or the artificial intelligence cannot do anything. You really need to know what you're looking at, because the volume of data is so immense that even the filtered variants may not really be of your gene of interest. So you have to have a thought process even to go through these filters and to go through the variants. And additionally, your AI may not pick out what you're looking for. I mean, recently I've solved a case where the AI didn't pick up even a pathogenic variant, though it was only one variant, because the phenotype had not reported that. Remember, all these filters are through what we call HPO terms, which is the human phenotype ontology. And you have to feed that for the AI work. So we are one of our DNB residents is doing a project of the traditional sequencing versus the AI in a same cohort of patients, and just work out how well each works. So
definitely.
There's a lot coming in, but I'm not sure that bedside the bedside phenotyping can be done away with, no matter how much of AI we have.
And Dr. Sharma, can I add to this one point, which is broader, which has really caught the storm, and in fact, any international conference you go to, it's one of the most talked about thing. It's about reanalysis of your data. So if you have a patient whereby you've done exome and you've done genome, and you're still not able to identify something, it simply means that you may have a variant in a gene of interest. The problem is that the gene has not been curated yet. So every couple of years, you see that you have OMIM and all of the databases, they go through massive series of upgrades. And because of that, you may able to carry out the diagnosis at that point. It has happened to us as well, that after 5-year gap, we finally managed to identify that the variant was sitting right there. The problem was that the gene wasn't very well curated. So if you have not been able to identify something, please do not be disheartened. It may very well be that the gene itself is not curated. So always keep a lookout at your data. We already have a lot of data. We just need to know how to analyze that data.
Definitely, I agree with you. For this point, I can say, in general, means what I believe, AI definitely needs intelligence, and that intelligence has to be given by the humans only, because it has not, it doesn't have its own brain. So whatever we feed, whatever we curate, the things will be keep on improving, and as a human population, everything will be surprised always, because there will be always new. So something always will be new for us. It's not like something that is we have like mastered everything. There will be always something new that we are not aware of, or something that will coming across for the first time for us. One thing that we discussed about, I think national policies are coming up. So anything that you would like to share on the national policies that are coming up by the government of India for managing such diseases, and the diagnostic and the treatment part?
So there is the National Rare Disease Policy, which came out in 2021. And it deals with two aspects. It deals with the aspects of increasing awareness related to diagnosis of patients and prevention of recurrence. And the second aspect it deals with is related to treatment of genetic disorders. And they have divided the categories of the genetic disorders into A, B, C. Disorders A are those that require a one-time treatment. So it could be related to a bone marrow transplant like you need for beta thalassemia or some of the immunodeficiency disorders. Then you have the category B, that is a lifelong treatment, but it's not very, very expensive. And these are predominantly the small molecule disorders or growth hormone therapies like you do it for Turner syndrome, or bisphosphonate therapies like it is for osteogenesis imperfecta. And category C are again genetic disorders which would require treatment, but the expenses for this are large, whether it's a
Mm.
one-time treatment or a long-time treatment. So within this come the typical lysosomal storage disorders that are amenable to treatment, would come the newer therapies that are related to treatment of spinal muscular atrophy and DMD, which are really very expensive. So this policy is there, and now the government is working out ways. They've announced 50 lakh rupees per patient, and they're still working out ways on how to reimburse patients for their treatment. So we hope in the near future. We've spent 2 decades in this field. I came into this field 2 decades ago, and 2 decades in this field, wondering if ever we would get a rare disease policy. So we are there, and I think in the years to come, in the times to come, we will definitely have guidelines that are there for early diagnosis, because remember, if you manage patients who already have a lot of disability, the management is not going to help. So you need early diagnosis, you need multidisciplinary care, which involves definitive as well as supportive management.
Yeah, thank you, Dr.
The government of India is setting up centers and centers of excellence and the kendras that Dr. Harsh already alluded to. So I'm not going to repeat that.
Thank you, thank you. So I think we have covered many aspects. So I think Dr. Harsh has already covered one aspect of the question is there by anonymous attendee about the errors caused by the DNA polymerase during PCR. How we would be explaining those errors and finding out like whether it is error or it is like some mutation. So in general, I think NGS is one of those stuff that definitely picks up in a big way, because we sequence the copies of those strands, and definitely the innovation is also happening in the polymerase aspects. We can say very high fidelity polymerases are available so that we can minimize the error that are incorporation error that are coming up, and you will be seeing that one thing that Illumina is promising now in the coming sequencing chemistry is a chemistry X that will be 3 times more accurate as compared to the current chemistry. It will have 2 times longer read length, and at the same time 2 times faster also. So within the same time, you can have more longer read length, and you will be able to have 3x more accurate data. So those promises are definitely, we can say, a lot of biologists, a lot of chemists, a lot of physicists are working in the background so that we can master the biology right now at the level what is needed now. And especially to my heart is the single cell genomics and spatial transcriptomics, which are giving answers to all those different questions that initially we didn't thought that we will be getting such tools to analyze each and every cell at the transcriptome of every tissues and the areas, and at a resolution which was required. And now people are seeing multiple genes at a time, multiple cells at a time. Mostly in the immunological disorders, I can say we are getting a very good answers to many questions of colitis. I was just going through that one of the paper that they found that a drug can be used as a treatment for colitis, which was previously for any other medication. So correlating the findings and giving that what we say treatment and the finding out the, we can say, diagnosis is the aspect of a clinician and the pathological lab, I can say, or the biochemistry lab, where people are finding the reasons of the disease, and the clinicians are, or the pharmacists are finding the cure of the disease or how to treat the patient based on the finding that we are getting. And yes, we are moving in those directions where we will be certainly able to help the people who are suffering from the rare diseases. And definitely there will be some like low cost solutions always there. We couldn't imagine everyone can the biochemical test less than 1,000 rupees. Now you can imagine everyone is able to get the biochemical test in less than 1,000 rupees, which was like previously was very expensive. Now people are able to afford such tests. Definitely we are seeing that generic tests also will be going in those direction, where the human genome project started from 3 billion and now within less than $1,000 we can have the human genome sequence for for any person in the world. And that will be the target is like how to make it $100 biolumina. But the roadmap, I think the things are ready, and these innovations are going to help in those directions to achieve the $100 genome. So with these words, I would like to thank both our speakers. This was very enlightening to everyone. And the session is recorded. I think we will put it on YouTube also. And people can go through. We will share the links also. We will share on our social media. And we will keep on organizing. We would like to have various aspects of the people in the rare diseases. And these events will be coming up. And you can see a QR code for the feedback of the webinar. And okay, so so some goodies are awaiting for all the people who will be like filling this form, and we'll be announcing those on our website through LinkedIn. So just put on your camera, scan it, and fill up the feedback form. So it'll stay for some time, but yes, I think, thank you very much for this wonderful talk.
Thank you. Thank you. Thank you all for having us here, and thank you all for listening.
Thank you so much. Thank you. And I'd really like to thank the delegates for giving a lot of time to the webinar. And
yeah, we had like almost 75